Bacteria replication and bacteria growth. This is my glossary. Binary fission. Binary meaning two and fission meaning splitting. Next is reproduce, to make copies of themselves. Next is Z-ring, a structure that squeezes the middle of a cell. A septum is a wall that takes the Z-ring's place that separates the two sides of the cell apart. Clones are cells that are identical copies of a cell that receive the same DNA. A culture flask, a flat plastic container used in laboratories. This is the other part of my glossary. A growth medium is a water-based liquid that is used to maximize the bacteria growth. And generation time is the time it takes for one cell to divide into two during the exponential phase. This is the steps to bacteria replication. A bacteria cell first makes its copy of its genetic material in a DNA molecule and passes on one copy to each side of the cell. Next, the cell stretches sideways and creates a structure called the Z-ring. The Z-ring then squeezes the center of the cell. After a wall, called the septum, replaces the spot where the Z-ring was to keep the two sides apart. Finally, the cell membrane disconnects to complete the cell division. One cell has become two. The new cells are called clones. Here's a picture that really helped me understand this concept. Bacteria growth. Bacteria used in laboratories are grown in flat plastic containers called culture flasks. The flasks contain a growth medium, which is a water-based liquid that includes sugars, proteins, salts, and other essential nutrients that bacteria needs to grow. The growth medium solutions in laboratories are designed to maximize the growth of bacteria. Bacteria growth in the laboratory follows the typical pattern. Let's take a look at these steps or phases. 1. The cell first takes up nutrients and makes proteins and DNA before dividing. This stage of the growth is called the lag phase. Now the cells are ready to enter the exponential phase, where the number of cells doubles with each cell division from 1 to 2 to 4 to 8 and so on. As the cells continue to divide, they use up all the nutrients in the growth medium. At this point, they enter the stationary phase, when cell division stops. Without a further source of nutrients, the cells then enter the death phase, and the number of living cells decreases. The time it takes for one cell to divide into two during the exponential phase is called the generation time. Hope you enjoyed.